I'll invite Rebecca Rooney up. So Dr. Rebecca Rooney is an associate professor at the University of Waterloo and carries out research in wetland ecology. She's a world expert on biomonitoring and wetland assessment. Her research supports implementation of wetland policy, invasive species management, and the protection of species at risk. Rebecca's pioneering work on the response of invasive Phragmites to herbicide-based control and the recovery of native plant and animal communities after Phragmites removal has been instrumental in supporting our efforts to control Phragmites in Ontario. So thank you, Rebecca, and I will pass it over to you. And I guess one quick note, please submit any questions you have for Rebecca at the Q&A just beside the chat, and we'll answer them at the end. So thank you, Rebecca. I'll let you take it away.
Thank you, Gabby. I'm going to share my screen here. And now I can't see Remo, but hopefully you all can see my slides and hear me. Is that right, Gabby?
It looks great. Thanks, Rebecca.
Perfect. Thanks. So I like to always start by thanking the partners without whom this work wouldn't have been possible, those who have funded our research and contributed to it, participated in it, and really worked together to help build this sort of frag fighting community, as well as the 22 plus trainees that I've managed to support and educate through this research. I also want to thank Dan and Mary Lou Smoke for the water song, thinking about our theme today, community resilience, a ceremony that has continued for 1,000 years seems like true resilience to me. So what a perfect way to kick things off today. But in the title of my talk, I've called The Future of Frag Control. And as we move forward into the future of frag control, behind, but I think it's helpful to reflect on where we've come from. So a brief history: if we had a time machine and we took it back to 2015, we'd see that frag was really plaguing our coastal marsh. This is a map of the distribution of Phragmites in 2015 in the Long Point Peninsula on the north shore of Lake Erie, where we've done a lot of research. And this was created by Ducks Unlimited, and it just shows the tremendous extent of the invasion at that time. Our hands were pretty tied because we didn't have a herbicide that we could use to control it in wet areas. We didn't have a product that was licensed for use over standing water in Canada, though our US neighbor Mazapir and glyphosate to manage frag really for years. So it had become very widespread and we had limited tools to do anything about that. We had these mechanical methods that for example, by my colleague Lynn Short, using the spading technique, which maybe some of you have used as well. These were effective, but worked under certain conditions. To cut to drown, you need the water to be high. Spading, you need the water to be low. Fire, we had many winters where the temperatures just didn't get cold enough to support doing controlled burns. So these techniques were effective, but they had their challenges. And if the efforts weren't sustained, we'd see frag come back. So just the scope of the problem was pretty disheartening. But we knew we had to do something. It was Paul Catling who first labeled Phragmites as Canada's first invasive plant. He did that in 2005, so a decade before that map that I showed you. And he called it that for good reason. For example, an analysis of over 200 species in Ontario that was done for the Ministry of Natural Resources and Forestry concluded were directly threatened by Phragmites. That was over 70%, 38% of our reptiles, over 30% of our fish. So it's affecting a wide diversity of species, especially species at risk. Repeating a survey that had been carried out in 2002, 2003, a student of mine back in 2014, Courtney Robichaud, observed that where frag had become established for, like a decade, so there was a reduction in avian abundance and a change in the composition of the bird community. In terms of that change, we saw there were fewer aerial insectivores like barn swallows, purple martin, tree swallows. There were also fewer large-bodied marsh nesting birds like bitterns. Phrag invasion also changes the nutrient stocks in marsh vegetation. So a student of mine, Sarah Yuckin, did a study on the composition of plant tissues, contrasting phrag with some of the dominant species that were native that phrag was displacing, things like cattail, Canada blue joint grass. And she concluded that invasive phrag leaves contained much higher amounts of nitrogen for every gram of carbon. So phrag invasion was also increasing the amount of phosphorus and potassium in leaf tissues. And this is affecting the whole ecosystem. For example, it was speeding up carbon cycling. We just published in the journal Wetlands, Ecology, and Management. And I want to pause here, actually, so I don't forget. If anybody would like copies of the papers that my grad students and I have published over the years, please get in touch with me and I can provide you a free copy. Please do not pay journals to access them online. We're more than happy to share copies with you. So please get in touch if you'd like one. So back to the science, thinking about carbon cycling, when phrag was replacing meadow, marsh, and emergent marsh plant communities, it doubled to tripled the canopy height, and it increased just the sheer mass of plant material. It reached as high as 3.3 kilograms per meter squared. That's 7 pounds of plant, more than you'd get in a cornfield that you were, treating with fertilizer and herbicide. So, there was a huge increase in the amount of carbon being sequestered, but it wasn't benefiting us in terms of climate change because that carbon was also being released much faster. So here, and I hope you can see my little cursor, here I have these pictures of leaf litter bags, and we used these to do an experiment where we measured the decomposition rate of the leaf litter. And we found, as you can see over time here, the mass of that leaf litter in Phragmites decreased much faster. In fact, it was decomposing twice as fast as the litter that came from cattail and Canada blue joint grass. And the reason that it was decomposing so much faster, we concluded, was actually because it had so much more nitrogen, phosphorus, and potassium than the native plants. And the microbes really gobble that up quickly. So compared to the litter that was being made by native plants, the litter being made by Phragmites decomposed much faster. And in this audience, most of you have stood in a patch, so you know how dense it is, how dark it is in there. Phrag invasion is really intercepting a lot of incoming light. It's lowering the temperature of soil and water. So the functioning of the coastal marsh ecosystem where phrag was invading was really being transformed. So with the scope of the impacts we were seeing from phrag invasion on species at risk, on birds, on basic ecosystem functioning, the Emergency Use Registration Phrag Control Pilot Project launched in Long Point and Rondo in 2016. And so far, they've treated over 1,500 hectares of invasive phrag using glyphosate-based herbicide. This was permitted under from the Pest Management Regulatory Authority with Health Canada, and it was the first ever overwater application of herbicide to control phrag in Canada. We used helicopters and ground-based methods to administer the herbicide, and most of the treated area then was either rolled or mowed to knock down the dead phrag stems, and then there was spot treatment to eliminate any regrowth or missed patches. That was done primarily with ground-based application. This project was really successful. First of all, we found there was a very low risk of exposure from glyphosate as a result of the herbicide application. We monitored water quality in treated areas for several years, the first several years of the Emergency Use Registration Project, and we never observed concentrations that approached thresholds of concern for aquatic life. And as you've no doubt seen me present before, the project was also really effective at suppressing Phragmites. So we saw a 99% reduction in the number of stems per meter squared in the control plots. So again, I'm going to pull my cursor over here. Hopefully, there it is. So this is before any treatment took place, contrasting the control plots where we were not going to spray herbicide with the treated plots where we did spray herbicide. That herbicide application took place in the fall of 2016, so after we closed. And right away, we had this 99% reduction in stem density Phrag compared to the control plots. But what was exciting was that really persisted at about a 95% reduction in stem density up until 2020, after which the project had been so successful, they treated our control plots in Long Point. And so we can't measure them anymore. But we were back there in 2022 sampling the treatment plots, and the extent of Phrag continues to be really small. So very successful at reducing Phrag stem density where we applied treatment. But the successes didn't stop there. This is also research from Courtney Robichaud. And I'd mentioned that aerial foragers like barn swallows were noticeably less abundant in areas where Phrag had invaded. That's here, this purple line, compared to the remnant habitat marsh where Phrag had not invaded, which is this yellow line. But interestingly, within just a couple of years of treatment, we saw aerial insectivore abundances increase in the treated area. So we saw pretty rapid recovery. And this was likely due to a big increase in the emergence of invertebrates in the early part of the summer from the areas where Phragmites was treated. And this was mainly from these non-biting midge larvae, which we call chironomids, and they're a great source of food for swallows. These are some photos of chironomids emerging and a dragonfly emerging. These were prime food for our aerial insectivore community, our swallows. We are also now seeing an expansion of swamp rose mallow, which is a species of conservation concern in treated areas in Big Creek. And we saw an expansion in endangered bent spike rush in the Long Point National Wildlife Area. So we're starting to see these benefits for species at risk from the Phrag suppression efforts. And for those who caught Megan Jordan's lightning talk last week, we're also seeing some improvements in the vegetation community in terms of its value for waterfowl forage. And if you don't know what I'm talking about, that's okay, because you can check out her poster on this when we have a break in Remo. Please go and visit Megan Jordan's poster. Earlier work that Megan participated in for her master's thesis showed that even though within the first few years after herbicide application, we had these sort of secondary invasive species take over, things like Eurasian milfoil, European frogbit. Here, that's these orange bars. Monitoring the plots through time, we saw initially this big invasion by those secondary invasive plants, but then after a few years, native plants, the green bars, did come to dominate the plots. And so that was, but this succession process, it's not guaranteed to go this way. For one thing, we were definitely aided by high water levels on Lake Erie that kept Phrag from reinvading. The yellow here shows you the entire period since the emergency use registration began. And it's above the long-term average lake level for Lake Erie. This spring, in the red, is kind of the forecast zone here. This is really the first time that we're going to drop below that long-term lake level average since 2015. So we're headed back into a low water period. And, we saw this coming, the writing on the wall. So we were nervous what was going to happen to these treated areas once the seed bank was exposed. And Megan Jordan's master's work included a greenhouse experiment to try to see what was going to grow up from the seed bank in the Long Point wetlands once the water levels went down. And she had these different treatments, some with a few inches of water, which we call flooded, and some which were just kept moist. So we had moist soil and flooded treatments. And what she observed was that in areas where the Phragmites was still growing, even a few inches of water was enough to keep the Phrag seeds from germinating. But that once that seed bank was exposed to the air, there's this huge pool here of Phragmites seeds ready to take off, up to 700 per meter squared. But reassuringly, in the areas that we had treated, like a year or two before, even in the exposed seed bank, the amount of Phragmites there ready to germinate was a lot lower, but there's still enough to present a reinvasion concern as the lake levels are coming down. This figure is from a great paper by Christine Rohal and Karen Kettenring in the Great Salt Lake wetlands in Utah, where they're doing a lot of really similar work. And it highlights some key assumptions that we're making about the recovery of vegetation after we suppress Phrag. when we're relying on the native seed bank to repopulate an area that we've removed Phrag from. And so these are sort of five main, there we go, five main assumptions. And the first one is that, the seed bank is going to be adequate to the task of repopulating the treated wetlands with native plants. So we know in more degraded areas where Phrag is more dominant, there aren't a lot of native species left, where Phrag has been established for a decade or more, the seed bank can become really depleted of native species. Whereas in less degraded areas, there may remain a lot of native plants in the seed bank ready to recolonize. The second assumption is that the native seed bank is going to be affected by our management actions. So maybe that rolling or burning is going to improve native plant recovery, or that one herbicide, might have more residual effects on emerging seedlings than another. The third assumption was that management is going to reduce the amount of invasive species in the seed bank, but not affect, this is the fourth assumption, the amount of native species in the seed bank. And finally, that the seed bank is even going to be predictive of what plants grow up in the field. So these are the sort of five assumptions that are implicit in what we're doing. How well do these assumptions hold up? Well, there's quite a lot of evidence that standing dead litter and the thick thatch that comes from rolling Phrag can delay plant recovery. Getting rid of that biomass is important to jumpstarting the seed bank, but even with the best strategies of removing Phragmites, high water levels can slow down the seed bank recovery. So we only have so much control over conditions that occur post-treatment. Megan's greenhouse experiment suggested, as I showed in an earlier slide, that the Phrag seed really is reduced by Phrag suppression with glyphosate. And at least in Long Point, she also showed, as illustrated here, that there's sufficient diversity of native plants in the seed bank to repopulate the areas where Phrag was suppressed. So this is those sort of third and fourth assumptions. You can see in the treated areas, there's still a lot of diversity of native seeds emerging in both the flooded and the moist soil conditions. But of course, Long Point is a relatively pristine area, and it's likely a best case scenario when we think about the health of the native seed bank in areas where we'd be controlling Phrag. If you think about areas where the seed bank is not so healthy, we might just not have the density or the diversity of native seeds we need to promote that native plant recovery. So you can imagine here in a place where we've treated in Long Point under the emergency use pilot project, there's ample sources of native plants to recolonize this little patch of Phrag that gets treated. Sure, when we did this, we observed some secondary invasive species as well, but these are pretty biodiverse native plant-dominated areas. In contrast, a lot of the places where we're trying to control Phrag are more like this. This is one of our study sites on the University of Waterloo campus that Claire Sean will be talking about later today. And it's across the street from a Sobeys parking lot. It's surrounded by housing developments. It receives stormwater runoff from the busy Fisher-Hallman Road. There isn't a lot of native wetland around to provide seeds, plant propagules to recolonize it. So instead, we can expect that there would be a lot of other invasive weedy plants around that could take advantage of the open habitat once Phrag is removed from somewhere like this. Native recovery following invasive plant suppression can be limited by insufficient native propagules in the seed bank, or from seed rain, or from large invasive seed banks that can facilitate invasive plants like secondary invasives, as well as reinvasion by Phrag. And thirdly, by inadequate environmental conditions for native plant establishment. So we need to be thinking, I think, a little more holistically about what happens after we remove Phragmites. Even where there are lots of native seed in the seed bank, and even where ultimately native plants were going to take over, like we saw in Long Point and Rondo, we first experienced those secondary invasions. And perhaps we could have expedited the transition to native plant dominated vegetation by seeding with native plant seeds. This is because of a phenomenon we call priority effects. So the main way that this happens is through something called niche preemption, where a niche is opened up by the removal of species, like we do with Phrag suppression. We remove Phrag from the niche it's occupying, making that niche vacant. And then any of the propagules or plant species present in the wider species pool in that region, in the seed bank, for example, are capable of filling that vacant niche. And the first one that gets there, it's kind of a scramble to fill that vacancy. Whichever species gets there first is able to more easily hold on to the niche, thereby sort of rebuffing other species that are trying to fill that same niche. So here the gray triangle gets there first, fills the triangle hole that's left by removing Phragmites, and these other species, the blue triangle, the yellow triangle, that are functionally kind of similar to that triangle hole, can't get in. So by seeding with a diverse mixture of native seeds after we control Phrag, we might be able to harness niche preemption to naturally resist reinvasion by Phragmites and secondary invasions. My PhD student, Jersey Fawns, attempted to test this in Long Point using a marsh organ experiment, which she'll tell you about later today. But briefly, she took seed bank samples from treated and untreated areas of marsh and grew them up under different experimental treatments in the field, in these things we call marsh organs that are like these pipes hammered into the marsh. And to some of these organs, she added native seeds, and to others, she didn't. And she added a combination of high and low diversity seed mixtures, and she sowed them at high and low seeding densities. And this is just a sneak peek at what she found, which is really exciting. She found that in both the high diversity treatment and the low diversity treatment, so this is the pink is the high and the blue is the low, it didn't really matter if it was seeding with a high density of seeds or a low density of seeds. The biomass of non-native plants in those unseeded control pipes that she didn't add any native plant seed to was double or triple that in the pipes that she did add the native seed mixture to. So what this means is that seeding, even with a low diversity seed mixture, even at a low density, can dramatically reduce the biomass of non-native plants that could come out of the seed bank and kind of undermine the restoration efforts we're making. But we need to scale this up, and we need to ensure that it works in larger plots. And we need to derive seed mixtures that will work in deeper water. So that's what Jersey will be working on this summer. Post-treatment seeding with native species to enhance recovery and resist reinvasion and secondary invasions, I think, is really the next frontier of Phrag management. And the next phase of questions are going to be about how to do this effectively. Like, how do we get the seed to stick to the soil in flooded conditions? How do we tailor the seed mixtures to provide natural resilience to climate change, to flood and drought cycles, or to stressful environments like that urban marsh that I showed you on the University of Waterloo campus? How do we maximize germination rates? What's the bare minimum density of seeds necessary for this? And what species are the real workhorses, the ones that are doing most of the heavy lifting, keeping out other invasive species? And which ones might be luxury items that we could include if we have unlimited budget? It's clear, I think, from our work that herbicide is a crucial tool that's going to let us more successfully manage Phrag across Ontario. But there are also new frontiers to push on how that herbicide gets applied. For example, there were concerns that track vehicles could harm turtles or snakes. And so we want to have tools that let us deliver herbicide precisely to hard to reach locations or in areas where we might fear for wildlife impacts. If we're not going in with track vehicles right now, our only option is really helicopters. And these have these really wide boom widths, like 4 meters. So even without herbicide drift or accidental overspray, which is depicted here from the Emergency Use Registration Project's first year of operation, in this great work that Adam Hogg did from the Ministry of Natural Resources and Forestry, you can see where the brown is where the glyphosate landed. And these sort of cyan colored tracks are where the helicopter was making its passes. Even without any of those kind of like accidental overspray or drip lines, there's going to be necessary non-target effects just because of the width of the boom. And we need to find ways to minimize that, because if you think about the native plants that are supposed to be recolonizing these areas, when we have non-target effects, we're landing that herbicide on the plant communities that we want to recolonize our treated areas. So we need ways to reach these remote, hard to traverse wetlands, to apply herbicide more precisely, to treat little patches before they grow, and to do it without harming wildlife. See if I can get this to go. This is my graduate student, Grace Lou Cole, and her thesis is investigating the potential for drone-based herbicide application in Phrag suppression under a research authorization from Health Canada. And she'll be presenting on it later today. But our hope is that her work can help us achieve more precise delivery with low impact and opportunities for spot treatment. This figure here is kind of illustrating that concept of how the smaller boom width alone can help us minimize non-target effects compared to the wider booms with the helicopter application. You can see the wider green polygons are the extent of Phragmites that we're trying to treat. And we don't want to leave any of it behind because we know when we do that, it's going to quickly recolonize the patch. But if our only ability to treat is using a wide boomed helicopter, you can see the amount of sort of overspray that's necessary to get those little edges because of the fixed boom width. With a smaller boom width on a drone, we can really trim back how much of that overspray has to happen to cover all of the Phrag. And minimizing non-target effects like this is really important for recovery because that neighboring adjacent plant community is where we're hoping the colonists will come from to repopulate these patches with native plants. The next steps in Grace's work include assessing how effective the herbicide administered by the drones is in suppressing Phrag and evaluating the accuracy of that drone-based application of herbicide. So she'll be talking more about that soon. Still, we don't want to be entirely dependent on herbicides for Phrag control. Thinking of our work with glyphosate again, even though we found it was quickly broken down in the water and the sediment, we did find that it bioconcentrates in these attached algal communities we call biofilm. You can see it growing here on one of our artificial substrates. And Verena Sesson and her colleagues at Trent found that it can accumulate in plant litter and leach back out into the environment. So we don't want to, be dependent on herbicide as our only treatment tool. We need additional tools that will supplement herbicides in more of an integrated pest management framework. And one of those possible tools is insect-based biocontrol. With classical biocontrol, it's all about reuniting invasive species with their natural enemies. Why is Phrag able to outgrow and outcompete native plants? Well, the leading theory is that, when it left Europe, it left behind its diseases, its pests and parasites, all of its natural enemies that helped hold it in check. With biocontrol of invasive species, the enemy of my enemy is my friend. And so this here is Claire Shawn, and she'll be reporting later on about her work with Michael McTavish and colleagues to study the efficacy of 2 moth species that have been approved for release in Canada to suppress Phrag. And last summer, we released the moth eggs and larvae at 2 sites near Waterloo, one that site I showed you earlier on Waterloo's campus. And we found that up to 77% of the stems were damaged by the eggs or the larvae that were released. She also found that the caterpillar feeding on Phrag impacted the growth, the physiology, and the reproduction of Phragmites, leading to shorter stems and overall less biomass, and importantly, to reduced flower production, which means that there's going to be less of that invasive Phrag seed going into the seed bank to challenge native plants in the future. Claire's going to revisit these plots next summer and continue to study the efficacy of the moss and the recovery of the native plant community, but the early results are extremely promising, so stay tuned for her presentation later today. To sort of sum up here, we've found that herbicide can be a powerful tool in Phrag suppression when we apply it over standing water, and it's an excellent and important new tool in our arsenal. In Long Point and Rondo, glyphosate application, it suppressed Phrag stem density by 95% or more over 5 years following application. But herbicide is non-selective, and it comes with non-target effects that endanger the plant communities we're trying to conserve. It can also impede recovery if we're eliminating these adjacent native plants that could have helped recolonize the treated patches. Drone-based herbicide application is a potential method that could minimize the extent of non-target effects and let us treat patches of Phrag that are small, isolated, situated in sensitive habitat, or that we can't safely drive a marsh master through. If drones turn out to be accurate, they might open up the ability to do spot treatments and safely catch any remnant Phrag that we miss or that recolonizes after that first treatment, without doing too much damage to the recovering native vegetation that we're trying to conserve. So Grace Lucole, over the next year or two, will be investigating just how accurate and precise drone-based application can be. But even with new application technologies that are more precise, we know herbicide alone is not sufficient to really tackle Phrag across the province. It's expensive, and repeated uses risk residue accumulation in sensitive wetlands. Ecology presents us two excellent additional tools that we're not really taking full advantage of. The first of those is biocontrol, and Michael McTavish and Claire Sean will be sharing some of their recent work on the two moths that have been approved for release in Canada. Biocontrol on its own won't eradicate Phrag from the region, but in combination with tools like herbicide, it can give us more versatility in Phrag management, and it can provide a gentler suppression pressure than herbicide that we hope will help shift the balance, allowing native species to compete and reestablish in our marshes alongside Phrag, maybe getting invasive Phrag to behave more like our native Phrag and start to play well with others. There's more that we can do to help our native plants compete with Phrag, though, and Jersey Fawns and her here in Ontario, and Karen Kettenring's group in Utah are looking at seeding as an integral part of the future of Phrag control. If our goal is to restore native marsh vegetation to support native biodiversity, we need to recognize that long-term Phrag invasion in less pristine places than our provincial parks and national wildlife areas where I've had the privilege of working, it's eroded the resilience of our marshes. Most places will not have the native plant diversity present in the seed bank to recolonize treated marsh once the Phrag is gone. We need to integrate reseeding into our standard operating procedures to make sure that we're not leaving a giant vacant niche that will just be filled by weedy or other invasive species. And if we're thoughtful about what we include in that seed mix, I think that we can harness priority effects to help our marshes naturally resist Phragmites. Last, the future of Phrag control really needs to be in continued partnerships between government and academics, environmental NGOs and stewardship groups, and individual landowners. We've made tremendous strides recently through this collective action approach. And as we begin to avail ourselves of these new tools and technologies to scale up Phrag suppression efforts here in Ontario, we'll need to continue working together. So the theme of today is community resilience. And really this comes from us, the energy we bring to this topic, and the support we lend each other. So let's keep it going. Thanks.
Thank you so much, Rebecca. That was excellent. We really appreciate that excellent overview of all the great research that's been going on. So exciting to see, and it helps us also look forward to some of the talks that'll be happening later in the day. So thank you very much. So we do have quite a few questions, and I know we're a little over time, so we'll try and motor through them as best we can, but some really great questions, so I definitely want to make time for this. And just to let you know, if you haven't submitted a question, you can do that through the Q&A, and you can also upvote questions that are there by just clicking to the left of the question, that up arrow. So first question, would mulching the biomass assist with native recovery versus rolling/burning? This could quicken decomposition.
Yeah, so we definitely found in Sarah Yuckin's work that decomposition of the sort of Phrag litter was sped up by knocking down the Phrags and making them wet. The key is to make them wet. So long as they're dry, they can stay standing for years, for 3 or 4 years. We've seen stems that have stayed standing. So getting them wet is important, but you want to do that in a way where you're not creating like a thatch that's going to impede the native seed bank from, germinating. Those little seedlings struggle to get through really thick litter layers. So burning is an excellent practice that can really help, not just with Phrag control, but with general, I think successional trends in our marshes. But it's not always safe, or it's not always possible. So other options like rolling and mulching can be really effective.
Awesome, thank you. Does the research of using a drone explore the stress of such a machine on the birds in the wetland?
So we have actually looked at the immediate effects of Phrag control on the bird community with work that my graduate student Marissa Zago completed. And we really didn't see much of an effect on the birds at all of the initial treatment. The work that Courtney Robichaud did was on, once the vegetation community starts to kind of recover, there are desirable changes in the bird community we hope to see, but immediately after treatment, we kind of don't want to see any negative effects, and we didn't really observe any. So I think it's a lot in the timing. The marsh masters are going in the fall. When the birds are done nesting, it would not be good to go rearing their young, but after they've fledged, when things are, the plants are still translocated, nutrients, so the herbicide is more effective at reaching the rhizomes, but it also, helps mitigate any impacts to wildlife like birds to do it in the fall.
Excellent. Oh, that's great. Would mulching the biomass assist with native recovery? Oh, sorry, that one's already been done. They move around a lot when people upvote. So similar to the biomass question, I think you sort of answered this, but just in case there's any additions, would a prescribed burn be warranted to reduce the extra biomass of decaying plant biomass? So I think you said that is a really excellent option. Great.
Yeah, I think when it's possible, it's a great.
Awesome. And what biomass removal techniques did Long Point use?
So in the emergency use registration, Rondo Provincial Park had hoped to do controlled burns, and they never really worked out. The conditions were never suitable to make it a safe undertaking. In Long Point, rather than using controlled burns, they'd wanted to do mowing and or rolling, and they did both, but I think the mowing was a bit more effective. The rolling tended to create the thicker thatch. Both of them were effective at knocking the stems down. Like I said, you want to avoid creating that really thick thatch that then the seedlings can't penetrate.
Okay, excellent. And do biocontrol options threaten native Phragmites? Right.
So I know that Mike McTavish is going to talk about that soon, and I don't want to steal his thunder, but I think the short answer is a lot of research went into ensuring that native Phrag wouldn't be harmed by the biocontrol agents before they were approved. So we don't think that they're going to negatively affect native Phrag.
Okay, excellent. And one more question before we jump to our break. Does the success you found dependent on treating Long Point sized patches?
That's a great question from an anonymous person. So, there was some really cool work out of Karen Kettenring's group that recovery was better when patches were small, which I think was a lot to do with, just how much native marsh there was to sort of support that recovery. The patches that we were looking at in Long Point and in Rondo were big. They were really extensive, as you saw in the map that I showed from 2015. And so, when I say we were looking at a best case scenario, I mean, because the context was really a natural landscape, there was a rich native diversity in the seed bank. But I think smaller patches, you'd expect even more success, especially if it's a small patch of Phrag, a halfway decent marsh that still has a lot of native plants in it. If you're looking at something like I showed on the Waterloo campus, where it's, basically a storm sewer that has got a lot of Phrag growing in it, there's not going to be a lot of native plant diversity in there. And so even though it's a sort of smaller patch, I think that it'll struggle to see native plant recovery.
Okay, excellent. Well, thank you very much, Rebecca. We really appreciate it. We're